Alright, in this video I want to go over the difference versus mitosis and meiosis. Now, when we said we went over meiosis is cell division that produces gametes. Gametes are also known as sex cells. If we went and looked at the cell cycle, we learned in the cell cycle that when we produce our own cells, in meiosis, we went from having 2N diploid to our sex cells, which have half the genetic material. So we say that this is a cell division that produces sex cells. It starts as a diploid cell, and we are going to produce 1N haploid cells. When we learned mitosis, we learned that this is cell division of the genetic material in the nucleus of cells. Now, this is going to produce body cells. Body cells are also known as somatic cells. Now, somatic cells are diploid, and when we divide, we're going to make more diploid cells. So a big difference between the divisions is this one makes body cells, this one makes sex cells. This one goes from being a diploid cell to making a diploid cell, meiosis, diploid, and then they become haploid. Now, we also learn in meiosis that we undergo 2 rounds of cellular division. So, in the first round, which we call meiosis 1, you divide your homologous chromosomes. You have crossing over all the things that we talked about in the last video, so I hope you're watching the video. You have 2N, you have your replication has already occurred. It's a diploid cell. Synapsis is when the 2 sister chromatids of the homologous chromosomes come close together. Crossing over is when you have a genetic exchange. But in meiosis 1, your homologous chromosomes, mom and dads, are going to separate. And after crossing over, you have 2 cells that look like this. Now, in meiosis 2, your sister chromatids are going to separate, and this creates 4 genetically different cells, which are all haploid. So, we literally can draw this out. Mom's chromosomes duplicate. Those are sister chromatids of moms. Dads are going to duplicate. They get close together. That's synapsis, and then you have crossing over. In crossing over, your dad's genetic material and your mom's genetic material of non-sister chromatids are going to separate, and then your homologous chromosomes separate. So in meiosis 1, you go from having 2N. Meiosis 1 is prophase 1, metaphase 1, anaphase 1, telophase 1. You go to having 1N, the homologous chromosomes separate. Now, these are called sister chromatids now because they're identical copies. So in meiosis 2, sister chromatids are going to separate. What you end up with is 4 genetically different 1N haploid cells. They're all genetically different because of crossing over. So, in meiosis 2, you say sister chromatids separate. Meiosis 1, homologous chromosomes separate. Now, when you produce body cells, there's only 1 cellular division. You're making body cells, which are 2N diploid. So, you've already had replication occur. So, you have your mom's and your dad's homologous chromosomes that are 2N. However, when you talk about this, these are going to make two genetically identical cells. There's only one cell division. So you go from having 2N, diploid, mom and dad, to having genetically identical cells. So these are the same, and these are all genetically different. These are making diploid cells. These are making haploid cells. So that is the difference between mitosis and meiosis. Basically, in meiosis, it's the same phases as mitosis, which is going to be PMAT. However, you would say prophase 1, anaphase 1, telophase 1, prophase 2, metaphase 2. Everything falls pretty much the same when you talk about the metaphase plate, the middle, or anaphase, but the only thing that's different is what you're pulling apart in meiosis versus mitosis is only pulling apart the sisters. So this is just an overview. I hope you're reading your textbook.